Hello, everybody. My name is Robert DiMartini, and in front of you is an exaggerated and generalized image of a human cell and all of its wonderful components. We're all very familiar with this image by now. All of these components play a role in its function. We know that, but I come to you with an opinion. I think I know which components are the most special. Today we're going to talk about how this cell reproduces, how it takes its shape, how it connects to other cells, how it maintains its shape and structure in resistance to natural stresses, and how it moves its organelles or keeps them anchored. These components responsible for these actions, right here, are none other than the cytoskeleton. Here, looking like a wad of macaroni, is the centrosome, or singular centriole. These are two centrioles. It's a centrosome. They produce microtubules. Here is an intermediate filament, this thicker one here. And these thinner boys are microfilaments. The cytoskeletal components, including the centrioles, are among the non-membranous organelles. There's no membrane present, as there is in mitochondria, the Golgi body, or the cell membrane itself. This is a microtubule. It's composed of a coiled chain of tubulin dimers. The chain is referred to as a polypeptide. It's essentially a chain composed of amino acids. That's right, it's an organic polymer, structurally similar to plastic. That is wild and fascinating. Centrosomes are essentially extruders of our microtubules. Here is a microfilament. Without a doubt, my favorite cytoskeletal component. Here's something to think about for later. Connect one of these to a Z-disc and place it next to myosin fibers, or fibers with myosin heads, and what do you get? More on that later, but you should already know, probably. A microfilament is a protein chain, much like a microtubule. A microfilament is composed of actin, an amino acid. It is formed by two polypeptide chains, which seem to resemble ocean waves, which is sort of nice. Here, finally, are keratin fibers, known as intermediate filaments. They're made of the same stuff that your hair and nails are composed of. It looks like a rope, and also in part functions like one. Here is a nice visual aid to explain the function of a microtubule. It's largely responsible for movement within the cell. I'd like to specifically talk about two types of movement, though. The movement, which occurs during mitosis, and the movement of cilia, as most commonly seen in columnar epithelial cells. Let's go, bronchial tubes. Mitosis, right here. We cannot do that without microtubules. As you can see, microtubules from polar ends of the cell latch on to kinetochores on this chromosome and pull them apart like a wishbone, if a wishbone broke symmetrically. Thus, genetic material is transported by a microtubule from one side to the other. And of course, our centriole or centrosome produces those microtubules. You can see them right here. So what if a cell doesn't mitose? As we touched upon in the lesson about nervous tissue, it would have no need for the centrosome, nor a microtubule of any kind, as it does not mitose and is not ciliated. Can you think of another human cell that has no centrosome or microtubule? Hint, hint, red blood cells. Here's Another beautiful model of a centriole, or I guess centrosome. Are they connected? I don't know. Creating cilia, another way which microtubules create movement. Notice that the centriole anchors itself here to the cell membrane. And here is how those cilia move. Just like a little wave in motion, like also cilia moving mucus. I'm assuming that this is a columnar epithelial cell in the respiratory system. That's my assumption. Here's a sperm cell. It contains a flagella, which helps it move, powered by microtubules. And here is a microfilament. In this picture, you can see a wobbly phospholipid bilayer, and under it, actin filaments. I think that this picture is meant to convey the shaping power of microfilaments. Underneath the cell membrane lies somewhat of a carpet of microfilaments which bolster the cell membrane and assist in the movement of stuff through the cell membrane. That is with the help of a little protein called myosin, which, hint, hint, is not only found in muscle cells. The myosin molecules and actin filaments make all kinds of movement within the cell. How do organelles swimming in an ocean of cytoplasm get from one place to another? Well, they may have a coating of actin's favorite molecule, myosin, and if not, another similar protein. And there's a bunch of them, but myosin stands out. In this case, the organelle, let's take a Golgi vesicle headed towards the cell membrane, for example, latches onto the microfilament because it's coated with myosin, latches onto the microfilament, and is pulled along, which I imagine creates a sweeping current, pulling things along with it, if that is the case. Another example that we are familiar with is muscular contraction. Remember back in the first part when I asked, what do you get when you attach a microfilament to a Z-disc and put it next to a myosin fiber? If you said muscle, you're absolutely correct. So here it is briefly. This is my favorite part. Another use of this wonderful component is to become a zip tie of sorts during cytokinesis. I'm not sure if myosin is involved here, but I'm going to make an educated guess that there is a protein or two which binds to actin, causing a ratcheting of that cell membrane surface shut. Because you remember the carpet of actin filaments is right under the cell membrane, drawing it shut like a drawstring bag. And in a similar fashion to the way organelles move and to the way muscles contract, I would imagine. There's probably a lot more that goes into it, but for the sake of this lecture. All right, finally, here is intermediate filament, made of the same stuff as hair. It is a simple structure with a simple purpose. I thought this slide explained it beautifully. As stated earlier, these fibers look and function like a rope. They anchor things, as opposed to microtubules and microfilaments, which both move things. The intermediate filaments create and maintain the cell's main structure, and also, as stated here, resist the compression of the natural world. Imagine your body having none of this. Your skin would be like Play-Doh, I assume. As a rope would, intermediate filaments can form cell junctions. So here, you can bond cells to other cells with these. All right, we talked about mitosis, but I wanted to give a visual to bring it all together, especially with our polypeptide friends. Microtubules and microfilaments. There's a lot going on here, so I won't get too much into the details. So here is prophase and prometaphase. What you see are centrosomes, membrane, and various organelles arranging themselves magically into shape. It doesn't mention it here, but I have a very, very strong suspicion that microfilaments have a lot to do with this. That carpet of filaments under the membrane and throughout the cell made it look like a football. I just have a feeling. The centrosomes move to polar ends, and like the kraken, spread out into spindles, headed towards the chromosomes needed to make a new cell. Now in metaphase, we are neatly arranged with everything hooked up, thanks to these filaments. And here we have anaphase. Everything's pulled apart and to opposite ends along the microtubules. We got telophase, where the microtubules get out of the way and microfilaments can clean up shop. And finally, our friend cytokinesis, where actin microfilaments work exactly as expected, in conjunction with binding proteins. The cytoskeleton is amazing. I hope something new was learned today and that you now have a new appreciation for tubules and filaments of all kinds. And I hope you and I share an opinion that the cytoskeleton is the most special part of any cell. Thank you.